Have you ever wondered how chemical reactions in your body happen so quickly and efficient? The answer lies in enzymes and their fascinating field of study called enzyme kinetics. Enzyme kinetics is the study of chemical reaction catalyzed by enzymes. It involves measuring the reaction rates and exploring how they change under different conditions. But what makes enzymes so remarkable? Enzymes are proteins with high molecular weight that react with specific substrate to produce one or more products. These biological catalysts are highly selective, each designed to interact with a particular substrate or reactant with incredible precision. Besides that, the relationship between substrate concentration and enzyme activity can be described using the Michaelis-Menten kinetics. This equation helps us understand how the reaction rate changes based on substrate concentration, initial velocity, and maximum velocity. In simple terms, enzyme kinetics reveals the elegant efficiency of biochemical reactions and provides the key insights into how life is sustained at a molecular weight.
The application of enzymes in food and beverage industry. Firstly, we have enzymes as a sweetener production. In this case, enzyme will help to convert the complex carbohydrate into a simpler sugar. Firstly, the amylase and glucoamylase will break down the starch into glucose during the production of glucose syrup. And then the glucose isomerase will convert glucose into fructose that creates a high fructose corn syrup, which is widely used as sweetener production in soft drinks and processed foods. The next one, we have enzymes in the baking industry. In this industry, enzymes are used to improve dough quality and enhance the texture. We have three enzymes used in this industry, which is amylase, protease, and xylanase. Amylase will breaks down the starch into sugars and promote the fermentation, and then the protease will modify the gluten proteins, which will improve the dough elasticity and reduce mixing time. And lastly, we have xylanase, which is strengthen the dough by breaking down the hemicellulose and increasing the stability of the dough. Lastly, we have enzymes in the dairy industry, which play a crucial role in enhancing product quality and efficiency. For the example of enzymes used in this industry is chymosin rennet, which is used in cheese production to coagulate milk proteins that will form curds.
Hi, I'm Marsha Kamarina. Next, enzyme also can use in forensic science. Studying poison and drug in forensic science involve analyzing how the body processes this substance using enzyme. Enzyme can break down drug and toxin in the liver at specific rate. By studying the kinetic of this enzyme, forensic scientists can determine how much of the substance was taken, how long ago it was ingested, and its effect on the body. For example, poison like cyanide inhibit critical enzyme which can be measured to confirm cause of death. This information help in cases of overdose, poisoning, and even detecting drug use in criminal investigation. Secondly, DNA testing in forensic science use enzyme to analyze genetic material from crime scenes evidence like blood, hair, or saliva. Enzymes such as Taq polymerase are used in polymerase chain reaction (PCR) to amplify tiny DNA sample, creating millions of copies of analysis. Restriction enzyme cut DNA at specific sequence, which help in creating DNA profile unique to individuals. These profiles are then compared to suspect or database to identify a match. This process is highly accurate even with degraded sample, making it crucial for solving crimes, identifying victims, and justify the innocent. Lastly, determining the time of death or post-mortem interval (PMI) involves studying the activity of enzyme in the body after death. Once a person dies, enzymes naturally present in tissue and cell begin breaking down the body, a process called autolysis. Forensic scientists measure the rate of this enzyme activity, which change predictably over time. To estimate how long someone has been murdered, factors like temperature, environment, and the type of tissue analyzed can influence enzyme kinetics. So these are also considered. This method is particularly useful when combined with other techniques, such as studying rigor mortis, to provide a more accurate timeline of death.
There are a lot of biological functions in enzyme kinetics, and one of it is enzymatic reaction. Enzymatic reaction work as catalysts in biochemical reaction. The picture given show how does it works. First, the enzyme has an active site where the substrate binds. The substrate fits into the enzyme like a key fits into a lock. Next, when the substrate is bound to the enzyme, a chemical reaction is triggered. This complex is temporary and the reaction is facilitated by the enzyme. The enzyme releases the product of the reaction, which are different from the original substrate. The enzyme itself is not consumed in the reaction and can be used again. Enzymes perform the critical task of lowering a reaction activation energy. That is the amount of energy that must be put in for the reaction to begin. Enzymes work by binding to reactant molecules and holding them in such a way that the chemical bond breaking and bond forming processes take place more readily.
Hi, my name is Aina. Now I'm going to talk about muscle contraction. What is muscle contraction? Muscle contraction is the physiological process that enables muscle to generate tension and produce movement. It involves the activation of tension-generating sites within muscle cells, primarily through the interaction of two types of protein filaments. The key enzyme that involved in muscle contraction is myosin ATPase. It catalyzes the hydrolysis of ATP and provides the energy that needed for myosin heads to bind and perform the power stroke. Pathways like glycolysis and oxidative phosphorylation will regulate ATP production during muscle activity. It will increase the enzyme activity to confirm that ATP supply to meet the energy demands of contraction. For the calcium regulation, the enzyme will regulate calcium ion levels within muscle cells. Also, it helps manage muscle fatigue by regulating the breakdown of ATP and recycling of ADP and AMP through enzyme like adenylate kinase.
In conclusion, for this topic, enzyme kinetics, we can conclude that studying this topic helps to improve understanding of rate of chemicals reaction that occurs in our daily life, important in both scientific and industrial application, most likely in the food and beverages industry. Also, studying chemical kinetics is crucial to enhancing product quality and transforming industrial application to a better future. Enzyme kinetics is widely used in industry like food and beverages, and also used in forensic science for DNA testing and toxicology analysis that could lead to a better future in medicine and also other industrial like food and beverages, and also botanical industry and also pharmaceutical industry. That's all, and thank you.